Hello students, when we have studied about the outer boundaries of the cell, we have read that in prokaryotic cell, the outer boundary is a three-layered complex structure, but it is not the case of eukaryotic cell, right? So in case of prokaryotic cell, this three-layered complex structure is called as cell envelope, but this cell envelope is not found in eukaryotes. So now in today's video, we are going to discuss about this cell envelope in detail. So when we talk about the cell envelope in a prokaryotic cell, it is made up of cell membrane, cell wall, and glycocalyx. Together, these three constitutes the cell envelope, which act as a single protective unit. Now, if we discuss the components of the cell envelope, the very first one from outside to inside is glycocalyx. So glycocalyx is majorly composed of polysaccharides, but sometimes protein is also a constituent. Now, its function is to protect the prokaryotic cell or bacteria from desiccation, as it is a very tightly bound or tightly knit matrix. So that is why it allows the bacteria, or it prevents the bacteria to survive through that inhospitable condition. Now, the second function that it helps the bacteria in attachment to the substratum. Along with that, it also provides the resistance against the host immune system. Now, if we talk about the types of glycocalyx, we can see that if it is present as a loose sheet, it is called as slime layer. But if it is present as a very thick and tough boundary, then it will be considered as capsule. After glycocalyx, the second layer will be the cell wall. So, cell wall is composed of peptidoglycans in eubacteria. Now, what is peptidoglycan? Peptidoglycan is basically a heteropolymer of N-acetyl glucosamine, also called as NAG, and N-acetyl muramic acid, also called as NAM. So, the glycosidic linkages makes the long chains of peptidoglycans, and they are further cross-linked by a short chain of amino acids. Now, what is its function? So, the function of cell wall is to provide structural support to the prokaryotic cell, and also it determines the shape of the cell. Now, on the basis of cell wall, we can classify the bacteria into two categories, and this particular classification takes advantage of Gram staining procedure. So, on the basis of Gram staining procedure, we can classify the bacteria into two categories. The very first one is Gram positive if the bacteria stains purple, and it will be considered as Gram negative if the bacteria stain pink. Now, why there is a difference in stain color? It is because in the cell wall of Gram positive bacteria, the peptidoglycan layer is quite thick. And in this particular case, the crystal violet iodine mix or complex, that means the primary stain and mordant complex, is unable to cross this boundary when the decolorization process is occurring. The decolorization takes advantage of alcohol. So during the alcohol rinse, this particular complex is unable to cross the boundary, and therefore the Gram-positive bacteria retains the stain and stains purple during this particular staining procedure. But in case of Gram-negative, as we can see that the peptidoglycan layer is quite thin. So in this case, the primary stain and mordant complex, that is crystal violet and iodine complex, can easily pass through the membrane during the decolorization procedure, which takes the advantage of alcohol. So that is the reason why it has to take the secondary stain, which is safranin, in case of Gram staining procedure. So this is the reason why these two bacteria shows different stain or different colors during the staining procedure. So now over here we can clearly see that in Gram positive, the peptidoglycan layer is quite thick, and in Gram negative, the peptidoglycan layer is quite thin. One more unique feature that we can visualize over here is that in Gram negative bacteria, there is an outer membrane, outer lipid membrane is also present, which makes this bacteria different from the Gram positive one. So this is how we are differentiating. So now let's move ahead to the third component of cell envelope, which is plasma membrane. Plasma membrane is composed of lipids, phospholipids, proteins, and carbohydrates. And they are structurally similar to the plasma membrane of eukaryotic cells as well. So these are the three components of cell envelope. For the detailed structure of plasma membrane, please go through the short video on plasma membrane. I hope you have understood all the components of cell envelope well. Thank you very much.